Good morning, dear students. Welcome to Nanagungutri online classes. In the last class, we were discussing about Human Genome Project. What is the main objective of Human Genome Project? Why Human Genome Project is called as Mega Project? And what is the methodology of Human Genome Project? And what are the silent features of a Human Genome Project? We have discussed in the last video. So, the most important findings during Human Genome Project was that the DNA of all the human beings are same up to 99%, and only 1% is different in every individual. That shows its identity. So today we are going to discuss a topic which is related on this point, that is DNA fingerprinting. Yes, so in DNA fingerprinting Human Genome Project on the result basis ಮೇಲೆ 99% human beings 99.9% all humans have same DNA. Same DNA means they have same set of nucleotides, sequences of nucleotides. 0.1% is totally different in different organisms. So there are 7 billion populations present all around the world. So all these 7 billion individuals have different DNA. That is by 0.1%. Ist percent ಒಳಗೆ 7 billion population human population ಒಳಗೆ ಎಲ್ಲಾದ DNA different. So what is this DNA fingerprinting technique? So it is a technique to identify the person based on DNA level. So based on this 0.1%, we can identify a person. In 1% particular individual identity. So this was given by Alex Jaffe. So he solved this technique. By the help of this technique, he solved a murder and rape case of 2 girls in Lancashire city in United Kingdom. So this technique, identification of particular person based on DNA, this technique is depends on DNA sequence. So if you know the DNA sequences, ಯಾವ 0.1% DNA sequence different ಇರ್ತೈತೆ ಅಂತ ನಿಮಗೆ ಗೊತ್ತಾಯ್ತಂದ್ರೆ you can easily identify the particular person. So this DNA level means DNA sequence basis ಮೇಲೆ ಏನು ಮಾಡಬೇಕು ನೀವು ಗೊತ್ತಾ ಮಾಡಬೇಕು. So all this human genome ಒಳಗ ಅತಿ ಹೆಚ್ಚು ಏನಿರ್ತೈತಿ ನಾನ್ ಕೋಡಿಂಗ್ sequences ಇರ್ತಾವೆ. ಏನಿರ್ತಾವ್ರಿ ಜಾಸ್ತಿ ನಾನ್ ಕೋಡಿಂಗ್ sequences. These sequences ಏನಿರ್ತಾವ್ರಿ tandemly repeated sequence ಇರ್ತಾವೆ. What type of sequence they are? Tandemly repeated sequence throughout the length of DNA. When DNA converts into chromosomes, DNA or chromosomes are converted ಗೊತ್ತಾಗ್ತೈತಿ ಆವಾಗ ಪರ್ಟಿಕ್ಯುಲರ್ ಕ್ರೋಮೋಸೋಮ್ ಟೆಲೋಮಿಯರ್ ಏರಿಯಾ centromere area, they are present on telomeres, they are present on centromeres, and they are present on heterochromatic area. So ಈ ರೀಸನ್ಸ್ ಒಳಗ ಅತಿ ಹೆಚ್ಚು ನಿಮಗೆ ಸಿಗ್ತಾವ್ರಿ ಟೆಂಡಮ್ ರಿಪೀಟರ್ ಸೀಕ್ವೆನ್ಸಸ್ ಸಿಗ್ತಾವ. ಇಫ್ you made a density gradient centrifugation, if you made density gradient centrifugation of DNA, the whole DNA is separated by the particular DNA that is tandem repeating sequences. This is called satellite DNA, and this is the whole bulk of DNA. So it forms a two peaks. One side the entire 99.9% ಇರುವಂತ sequences separate ಆಗ್ತಾವ, ಇನ್ನೊಂದು 0.1% ಇದ್ದಿದ್ದ sequences separate ಆಗ್ತಾವ. So ಇದಕ್ಕೆ ಏನಂತೀವಿ ನಾವು ಅಂತ ಟೆಂಡಮ್ ರಿಪೀಟೆಡ್ ಸೀಕ್ವೆನ್ಸ್ ಏನಂತೀವಿ ನಾವು ಸ್ಯಾಟಲೈಟ್ DNA ಅಂತ ಕರಿತೀವಿ. So these satellite DNA are two types. One is called mini satellite DNA, another one is called micro satellite DNA. So this satellite DNA contains repeated sequences. Based on number of repeated sequences, satellite DNA is divided into mini satellite as well as micro satellite. So what happens in a micro satellite? If sequences are less than 10, if the repeated sequences are less than 10, it is called microsatellites. If the repeated sequence they are 10 to 100, then it is called mini satellite. So DNA fingerprinting, so we use such type of repeated sequences, and those repeated sequences in mini satellite, they are called variable number of tandem repeats, VNTR. In mini satellite, those repeated sequences are called VNTR. So these are particular VNTR are probably, so they contains 11 to 60 base pairs. 11 to 60 base pairs are particular present in these VNTR. So based on these variable number of tandem repeats, we can identify the person, we can know the identity of a particular person. Okay, so these VNTR, they inherited from father as well as mother. So 50% of variable number of tandem repeats are inherited from father and 50% from mother. So that is why they are halfly shared with both the parents. On that note, we can also identify the particular child of a particular parents. Okay, so this technique also helps to identify children of a particular parents. Okay, because VNTR, variable number of tandem repeat, these sequences, adenine, guanine, thymine, cytosine, these sequences, they are inherited from our parents. So what happens during these techniques? How this fingerprinting, DNA fingerprinting works, and how we can identify particular person based on our DNA. Let's see the technique behind this DNA fingerprinting. If in a particular situation any crime scene, ಯಾವುದೇ crime scene ಒಳಗ ಮರ್ಡರ್ ಆಗಿರಬಹುದು ಅಥವಾ ರೇಪ್ ಆಗಿರಬಹುದು ಅಥವಾ ಏನಾರ ಆಕ್ಸಿಡೆಂಟ್ ಆಗಿರಬಹುದು, ಸೊ ಅವಾಗ ಏನು ಮಾಡ್ತೀವಿ ನಾವು, ಕಲ್ಪ್ರಿಟ್ಸ್ ಸಪೋಸ್ ಇಟ್ ಗಾಟ್ ದ ಸೋರ್ಸ್ ಆಫ್ DNA ನಿಮಗೆ ಸಿಕ್ತಂದ್ರ DNA ದ ಸೋರ್ಸ್ ಹೇರ್ ಸಿಗ್ತಾವ್ರಿ, nails ಇರ್ತಾವ, blood ಒಳಗೆ WBC ತಗೋರಿ, or semen samples or saliva, whatever it is, a source from where we can extract the DNA. Okay, say for example we got a source, this is a cell of a particular source. So first step is the collection of sample. First step is the collection of sample. DNA fingerprinting or samples we collect. So you have collected the particular sample. Now what you have to do is you have to isolate this DNA. You have to isolate the DNA. You have isolated the DNA by adding number of enzymes. Now the next step, this is the second step. Next step is cutting off DNA by restriction endonuclease enzyme. So DNA is cut, double stranded DNA is cut into different length. Okay, now you have to separate these DNA based on length. Cutting with next step, separation of DNA segments. Separation of DNA segments. So this is a third particular step. For separation of these DNA segments, what you will use? A technique called electrophoresis. So what happens in electrophoresis? In particular electrophoresis, you add here agarose gel. So ಈ agarose gel ಒಳಗೆ ಏನು ಮಾಡ್ತೀವಿ ನಾವು, DNA ಗೆ ಏನು ಮಾಡ್ತೀವಿ, ಲೋಡ್ ಮಾಡ್ತೀವಿ. agarose gel ಜೊತೆಗೆ ಏನು ಮಾಡ್ತೀವಿ ನಾವು, alkaline solution add ಮಾಡ್ತೀವಿ. ಡಬಲ್ ಸ್ಟ್ಯಾಂಡರ್ಡ್ DNA ಹಾಕವ್ರಿ, ಸಿಂಗಲ್ ಸ್ಟ್ಯಾಂಡರ್ಡ್ DNA ಹಾಕಿವಿ. ಎಸ್, ಸಿಂಗಲ್ ಸ್ಟ್ಯಾಂಡರ್ಡ್ DNA ಹಾಕಿವಿ. ಸೊ ಈ ಸಿಂಗಲ್ ಸ್ಟ್ಯಾಂಡರ್ಡ್ DNA agarose gel ಒಳಗ ಮೂವ್ ಆಗ್ತಾವ. ಎಲ್ಲಿ? ಅಪೋಸಿಟ್ ಚಾರ್ಜಸ್. ನೆನಪಿಟ್ಕೊಳ್ಳಿ, DNA ನೆಗೆಟಿವ್ ಚಾರ್ಜ್ ಇರ್ತದೆ. ಸೊ ಯಾವಾಗ ನೀವು ಕರೆಂಟ್ ಅಪ್ಲೈ ಮಾಡ್ತೀರಿ, ಇದು ಪಾಸಿಟಿವ್ ಚಾರ್ಜ್ ಕಡೆ ಹೋಗ್ಬೇಕು. ಅಲ್ಕಲೈನ್ ಸೊಲ್ಯೂಷನ್ ಏನು ಮಾಡೈತಿ, ಡಬಲ್ ಸ್ಟ್ಯಾಂಡರ್ಡ್ DNA ಫ್ರಾಗ್ಮೆಂಟ್ಸ್ ಸಿಂಗಲ್ ಸ್ಟ್ಯಾಂಡರ್ಡ್ DNA ಫ್ರಾಗ್ಮೆಂಟ್ ಒಳಗೆ ಕನ್ವರ್ಟ್ ಮಾಡೈತಿ. ನೆಕ್ಸ್ಟ್ ಕರೆಂಟ್ ಅಪ್ಲೈ ಮಾಡಿದ ತಕ್ಷಣ DNA ನೆಗೆಟಿವ್ ಚಾರ್ಜ್ ಇರೋದು ಕಾರಣಕ್ಕೆ ಪಾಸಿಟಿವ್ ಚಾರ್ಜ್ ಹೋಗ್ತೈತಿ. ಸೊ ಹೇಗೆ ಹೋಗ್ತೈತಿ ಅಂದ್ರೆ, the segments which are so small, they run faster on agarose gel towards the positive side. The DNA which has large strands, DNA segments have large segments are ಏನಪ್ಪಾರಿ ಹಿಂದ ಉಳಿತಾವೆ. So this separation DNA ಏನಾಗ್ತೈತಿ, ಸಪರೇಟ್ ಆಗ್ತೈತಿ. ಎದರ ಮೇಲೆ ಸಪರೇಟ್ ಆಗ್ತೈತಿ, agarose gel. Next, if DNA is in less quantity ಸಿಕ್ಕುವಂತ ಸೋರ್ಸ್
ಇದ್ರೊಳಗೆ ಕಡಿಮೆ DNA ಸಿಕ್ಕಿತ್ತಂದ್ರ ನೀವೇನ್ ಮಾಡಬಹುದು, ಇದಕ್ಕೆ ಆಂಪ್ಲಿಫೈ ಮಾಡಬಹುದು PCR ಟೆಕ್ನಿಕ್ ಮುಖಾಂತರ, ಪಾಲಿಮರೇಸ್ ಚೈನ್ ರಿಯಾಕ್ಷನ್ ಟೆಕ್ನಿಕ್ ಮುಖಾಂತರ ನೀವು ಇದರದ ಥೌಸಂಡ್ಸ್ ಅಲ್ಲ ಮಿಲಿಯನ್ಸ್ ಟುಗೆದರ್ ಕಾಪೀಸ್ ನೀವು ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಬಹುದು. ಸಿಗುವಂತ DNA ಸ್ವಲ್ಪ ಇರ್ತದೆ, ಇಲ್ಲ ಜಾಸ್ತಿ ಇತ್ತಂದ್ರೆ ದೇರ್ ಇಸ್ ನೋ ನೀಡ್ ಆಫ್ ಅ PCR. Okay, the next step after separation, the fourth step is southern blotting technique. Southern blotting technique. So ಈ southern blotting technique ಒಳಗೆ ಏನಾಗ್ತೈತಿ, ಸಪೋಸ್ ದಿಸ್ ಶೀಟ್ ನಾ ವರ್ಟಿಕಲ್ ಡೈರೆಕ್ಷನ್ ಒಳಗೆ ತೋರಿಸ್ತೀನಿ, ಈ ಶೀಟ್ ಈ ಅಗಾರೋಸ್ ಜೆಲ್ ವರ್ಟಿಕಲ್ ಡೈರೆಕ್ಷನ್ ಒಳಗೆ ತೋರಿಸ್ತೀನಿ, ಇದು ಅಗಾರೋಸ್ ಜೆಲ್ ಐತಿ, ಇಲ್ಲಿ ಏನಾಗೈತಿ ಲಾರ್ಜ್ small 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 DNA separate ಆಗಿರುತ್ತೆ. Southern blotting technique ಒಳಗ you have to choose particular sheet. ಏನಂತ ನಾವು nylon sheet, nitrocellulose paper. ಈ nitrocellulose paper ಗ ಅಗಾರೋಸ್ ಜೆಲ್ ಮೇಲೆ ಏನು ಮಾಡಬೇಕು, ನೀವು ಇಡಬೇಕು, ಕಂಪ್ಲೀಟ್ ಅಗಾರೋಸ್ ಜೆಲ್ ಮೇಲೆ ಏನು ಮಾಡಬೇಕು, ಇಡಬೇಕು. ನೆಕ್ಸ್ಟ್ ಇದ್ರ ಮೇಲೆ ಏನು ಮಾಡಬೇಕು, ನೀವು ಒಂದು ವೇಟ್ ಇಡಬೇಕು. ದಿಸ್ ಈಸ್ ವೇಟ್. Nitrous or nylon sheet paper or nitrocellulose paper absorbs the DNA bands which are separated on agarose gel. Agarose gel ಒಳಗಿರುವಂತ ಈ ಸಪರೇಟ್ ಆಗಿರೋ DNA strands ನೈಟ್ರೋಸೆಲ್ಯುಲೋಸ್ ಪೇಪರ್ ಒಳಗೆ ಏನಾಗ್ತವ್ರಿ, ಪ್ರಿಂಟ್ ಆಗ್ತದೆ. ಈ ಪ್ರಿಂಟಿಂಗ್ ಏನು ಮಾಡೋದು, ತಗೋ. ಯು ಕೆನಾಟ್ ಸಿ ವೇರ್ ಎಕ್ಸಾಕ್ಟ್ ದೇ ಆರ್ ಪ್ರಿಂಟಿಂಗ್. ಎಲ್ಲಿ ಪ್ರಿಂಟ್ ಆಗ್ಯಾವತ್ತು ನಿಮ್ಗೆ ಗೊತ್ತಾಗಲ್ಲ, ಬಿಕಾಸ್ ದೇ ಆರ್ ಇನ್ವಿಸಿಬಲ್. ಓಕೆ, ದೆನ್ ದ ನೆಕ್ಸ್ಟ್ ಸ್ಟೆಪ್ ಈಸ್, ದ ಫಿಫ್ತ್ ಸ್ಟೆಪ್ ಈಸ್ to choose VNTR probes. Variable number of tandem repeats ಇವು ಕೂಡ ಇರುತ್ತವೆ, ಡಬಲ್ ಸ್ಟ್ಯಾಂಡರ್ಡ್ ಇರ್ತವೆ, ಇವು ಕೂಡ ಇರ್ತವೆ, ಡಬಲ್ ಸ್ಟ್ಯಾಂಡರ್ಡ್ ಇರ್ತವೆ. ಇದರೊಳಗೂ ಕೂಡ ಏನು ಮಾಡಬೇಕು, ನೀವು ಅಲ್ಕಲೈನ್ ಈ VNTR probes ಅಲ್ಕಲೈನ್ ಮೀಡಿಯಂ ಒಳಗೆ ನೀವು ಸ್ಪ್ರೆಡ್ ಮಾಡಿ ಹಾಕ್ರಿ. ಇವೇನಾಗ್ತಾವ ಸ್ಟ್ಯಾಂಡ್ಸ್ ಸಿಂಗಲ್ ಸ್ಟ್ಯಾಂಡರ್ಡ್ VNTR probes ಏನಾಗ್ತಾರೆ, ಸಿಂಗಲ್ ಸ್ಟ್ಯಾಂಡರ್ಡ್. So millions together VNTR probes ಇವು ತಗೊಂಡು ಮಾಡಿ ಏನು ಮಾಡೋದು, ಈ ಶೀಟ್ ಮೇಲೆ ಏನು ಮಾಡೋದು, ಸ್ಪ್ರೇ ಮಾಡೋದು. ಈ ಶೀಟ್ ಮೇಲೆ ಏನು ಮಾಡಿದ್ರೆ ಸ್ಪ್ರೇ ಮಾಡೋದು. Remember, VNTR probes are identified in fluorescent dyes. Fluorescent dyes ಒಳಗೆ ಐಡೆಂಟಿಫೈ ಮಾಡೋದು. ಸೊ ಶೀಟ್ ಮೇಲೆ ಏನಾಗ್ತೈತಿ, ಸ್ಪ್ರೇ ಆಗುತ್ತೆ. The VNTR probes which have complementary sequences. Suppose particular VNTR probe the complementary sequence ಇದೆ ಐತಂದ್ರೆ, because VNTR probes are nothing but repeated sequences, and the DNA which is extracted here in these sheets, they also contains a repeated sequences. Old DNA or major part ಏನಿರ್ತದೆ repeated sequences ಇರ್ತಾವೆ. So same repeated sequences ಮತ್ತು VNTR ಒಳಗಿರುವಂತ repeated sequences complementary ಇರ್ತಾವೆ. So ಯಾವುದು DNA fragment repeated sequences complementary ಇರ್ತಾವೆ, particular probe ಆ probe ಏನಾಗ್ತದೆ ಹೋಗಿ ಅಟ್ಯಾಚ್. So it attaches. Complementary ಸಿಕ್ಕಿಲ್ಲ, ಅಟ್ಯಾಚ್ ಆಗುತ್ತೆ. The remaining probes they have to remove ಮಾಡಬೇಕು, ವಾಶ್ ಮಾಡಬೇಕು. ಪರ್ಟಿಕ್ಯುಲರ್ ಏನು ಮಾಡಬೇಕ್ರಿ, ವಾಶ್. ಆಲ್ಕೋಹಾಲ್ನಿಂದ ಪರ್ಟಿಕ್ಯುಲರ್ ವಾಶ್ ಮಾಡಿ ಬರೀ ಏನು ಉಳಿತಾವ್ರಿ, VNTR probes ಅಷ್ಟ ಉಳಿತಾವ, along with the person's repetitive sequence DNA. Now last method is pass this film on X-ray. X-ray auto radiograph ಅಂತೀವಿ. ಸೊ ಎಲ್ಲೆಲ್ಲಿ ಪ್ರೋಬ್ ಅಟ್ಯಾಚ್ ಆಗಿರ್ತೈತಿ VNTR ಪ್ರೋಬ್ ಅಲ್ಲಿ ಏನಾಗಿರ್ತೈತಿ, ಡಾರ್ಕ್ ಬ್ಯಾಂಡ್ಸ್ ಕಾಣೋದು ಸ್ಟಾರ್ಟ್. So the appearance of these bands are different in different So this band's appearance is different by 0.1%. Suppose the DNA which you get on a crime scene has these bands, and a DNA of a person on which you have doubt have the same bands arrangement. These VNT are used as a selectable markers. Okay, so same bands but the same DNA is present. Remember, DNA never change. Age to age, tissues to tissues, particular life cycle ಒಳಗೆ ಅಥವಾ ಯಾವುದೇ ಕಾರಣಕ್ಕೆ DNA ತಮ್ಮ ಪ್ರಾಪರ್ಟಿ ಕಳ್ಕೊಳ್ಳೋದಿಲ್ಲ, ಚೇಂಜ್ ಆಗೋದಿಲ್ಲ. ಸೊ ಸೇಮ್ DNA ಒಂದೇ DNA ಈ ಬ್ಯಾಂಡ್ಸ್ ಏನಾಗ್ತಾವ್ರಿ ಬರ್ತಾವೆ. ಸೊ ಪೇರೆಂಟ್ಸ್ DNA ಕೂಡ ಏನಾಗ್ತೈತಿ 50% ಪೇರೆಂಟ್ಸ್, 50% ಫಾದರ್ ಮತ್ತು 50% ಮದರ್ ಏನಾಗ್ತೈತಿ ಬ್ಯಾಂಡ್ಸ್ ಇಲ್ಲಿ ಮ್ಯಾಚ್ ಆಗ್ತಾವೆ. ಬಿಕಾಸ್ VNT are inherited from parents. Okay, so this is a technique based on which we can identify the person's identity. So what is the application of this DNA fingerprinting? So application is that we can identify a particular children's of parents. It can parents disputes. So they can identify their children. Yes or no? If any person claims that a particular child belongs to them, then they will do DNA fingerprinting technique. If in a big accident, burn or pieces, parts are there, we can join all the parts of a same person by help of DNA fingerprinting. Okay, you can identify the murderers or rapists if the source of DNA is present on them. Okay, so we can identify the country disputes also, because these 0.1% DNA shows some similarity towards a particular gene pool in which the species are sexually active. Means particular based on the region, the human population have different genome. We can also identify that points based on these DNA fingerprinting. So nowadays DNA fingerprinting is also used to detect the number of genetic disorders, or to particular identifies the presence of alien or foreign DNA. Like say for example, if a person is attacked by a viruses or any other bacteria, so on that DNA fingerprinting also we can use to detect or diagnose some kind of viruses. So this is about the DNA fingerprinting. So this is the last concept of molecular based inheritance. Tomorrow we will go with a new chapter. Okay, thank you.